Good morning to all of you. The today's new concept related with bacteria, that is the bacterial transduction. Under bacterial transduction, we'll see the generalized transduction, specialized transduction. First of all, we'll see the definition of bacterial transduction. That is the phenomenon of transfer of portion of DNA from one bacterium to another by a bacteriophage. It is called as a transduction. This process was first studied by Zinder and Liderberg. For studying that concept, he utilized a Salmonella typhimurium. He ran that experiment in 1952. Such a bacteriophage particles developing in a one bacterial strain often incorporate a piece of bacterial chromosome into their structure. If such a bacteriophage particles are known allowed to infect a different genetic strains of same bacterial species, they may transfer these genes to some bacterial cells which survive their infection. Such a transferred genome becomes a part of chromosomal material of a new bacterial strain. This new strain now contains some characteristic of the first bacterial strain upon which the bacteriophage was first grown. This indicates that the bacteriophages are responsible for transfer of genetic material from one bacterium to another bacterium. The Zinder and Liederberg in 1952 performed a YouTube experiment in such way there exists no possibility of a direct cell contact. They grow the two auxotropic strains in two arms of YouTube. Both the strains were physically separated by a sintered glass filter impermeable to bacteria. The recombinant still produced, it becomes smaller than bacteria, that is the bacteriophages. The phenomena of transduction is confined not only a transfer of chromosomal DNA, plasmid and episomes may be also transduced. First of all, we will see the generalized transduction. In this transduction, the bacteriophage carry the segment of chromosome which have more than one gene. The genes may be very closely linked to each other. In a case, the segment of donor chromosome is transferred when bacteria infected with a virulent bacteriophage. They reproduce with the bacteria and they kill a bacteria. Bacteriophages causes a lysis are termed as a lysogenic phages, and those which cannot show a lysis are termed as a template phages or prophages. The bacteriophages accidentally carries the genetic material of bacteria and not of its own. Thus, the bacteriophage is a biologically inactive. It again infect another bacteria and inject a genetic material. This material may contains many genes, so called as a generalized transduction. The second transduction, that is the localized transduction. In this transduction, the specific locus, that means the gene of donor chromosome, is transferred. The phage is important. For example, of localized transduction, this phage is able to transfer only galactose locus, that means the gal plus of E. coli bacteria, to another bacteria. When a single gene or locus is transferred, it is called as localized transduction. The bacterial transduction is the phenomena in which the bacterial genes are transferred from one bacteria to another bacteria with the help of bacteriophage. This happens when bacteriophage accidentally carries host chromosome instead of its own chromosome. This bacteriophage introduces a chromosome into another bacterium. This chromosome or segment will recombine with a host chromosome and bring out a recombination. Bacteriophage carries a very small portion. They are biologically inactive because they are totally missing its own genetic material. However, it is capable of infecting and attaching another bacterium. Thank you.